Hello friends, good morning. Today we'll discuss the second part of the video of the pre-nucleotide synthesis. So in the earlier video we discussed de novo synthesis. In today's video we'll discuss salvage pathway and the regulation of pre-nucleotide synthesis. So I will start with the salvage path in details first. So during the turnover of nucleic acids, the three basic purines are formed. That is number one adenine, second is guanine, and third is hypoxanthine. They can be directly converted into, for example, we have adenine, it can be directly converted into AMP, guanine can be directly converted into GMP, and hypoxanthine can be directly converted into IMP by this salvage pathway, right? So, first of all, we are having adenine, it is 6 amino purine, guanine is 2 amino 6 oxy purine, and hypoxanthine is 6-oxypurine, right? So first of all, we start with adenine. Adenine can be converted into adenosine monophosphate by the enzyme adenine phosphoribosyl transferase. For this reaction, we require phosphoribosyl pyrophosphate, and it can be converted into pyrophosphate, and we will get the compound adenosine monophosphate, right? So in the similar manner, guanine is 2 6 oxypurine by the enzyme HGPRT, hypoxanthine guanine phosphoribosyl transferase. With the involvement of phosphoribosyl pyrophosphate, it can be converted into guanosine monophosphate. So the phosphate is attached here. Similarly, we have hypoxanthine is 6-oxypurine. When you replace 6 amino with 6-oxy, you will get hypoxanthine, right? Hypoxanthine by the same enzyme hypoxanthine guanine phosphoribosyl transferase will form IMP, right? So the same pattern we require phosphoribosyl pyrophosphate, and it is converted into pyrophosphate, right? So this is energy saving pathway, because in de novo synthesis we require 11 steps and then we'll get IMP. But in this pathway we can directly synthesize this monophosphates from adenine, guanine, and hypoxanthine, which are produced in normal turnover of pre-nucleotides, right? And there is one disease linked to this enzyme HGPRT. The deficiency of HGPRT and transferase causes Lesch-Nyhan syndrome, right? Now we'll start with regulation of the pre-nucleotide synthesis. Now we'll start with regulation, right? There are three basic steps where regulation take place. First is the first step of de novo synthesis, that is the enzyme PRPP synthetase. Then the second step is glutamyl aminotransferase, and the third is the 11th product, that is from this IMP, right? So first of all, we'll start with the first enzyme PRPP synthetase. Ribose 5-phosphate is the precursor, and it is first converted into PRPP, phosphoribosyl pyrophosphate, by this enzyme PRPP synthetase, right? So this enzyme is allosteric enzyme, right? And it is inhibited by AMP, ADP, GMP, and GDP, right? PRPP is further converted into phosphoribosylamine in the second step by the enzyme PRPP glutamyl aminotransferase, right? So this enzyme second is under feedback control, right, by the relative concentration of AMP and GMP. Now the third regulatory step is IMP. After 11 steps of de novo synthesis, we will get IMP, right? IMP can be further converted into AMP, by this enzyme adenine succinate and GMP by IMP dehydrogenase, right? They, when there is sufficient concentration of both these, they can inhibit this by feedback mechanism, right? So that finishes with our regulation. There are 3 steps. Firstly, the first 2 enzymes, that is PRPP synthetase, and the second is PRPP glutamyl aminotransferase, and final regulation is at the 11th step when we'll get IMP and its conversion to EMP and GMP. They both control the synthesis by feedback mechanism, right? So that's now that finishes with our regulation and the salvage pathway. So the purine nucleotide synthesis, the 2 paths, de novo synthesis discussed in previous video and salvage pathway discussed in this video along with regulation. So keep revising well. Do like the channel and subscribe the channel. Keep studying. God bless. Thank you.